next speaker is Holly, who obtained her PhD at Curtin University in Australia in atmospheric iron fertilization. Did their postdoc at the British Arctic Survey in Cambridge, studying the ice core nitrate isotopes as a proxy for past UV radiation, and is currently at Rutherford postdoctoral fellow at the ARC in New Zealand, and is studying the development of ice core biomarkers. Holly's research aims to reconstruct past climate by investigating the present day sources and processes of chemical impurities preserved in ice cores in order to interpret longer ice core records. As part of a Rutherford postdoctoral fellowship, she is currently reconstructing past primary production using ice core biomarkers in the Ross Sea region of Antarctica. She uses aerosols, snow, and ice cores collected from a variety of environments, from subtropical savanna to the remote Southern Ocean and the Arctic Plateau, to understand transport, deposition, and archival processes of ice core proxies. So Holly, whenever you're ready.
Tēnā koutou katoa, and thank you for the invitation to speak at the Ice Core Young Scientist Seminar Series. So I'm going to continue with the theme of the modern atmosphere and talk about modern day aerosol observations over the Southern Ocean and how we can use these to help us in the development of new ice core proxies. I'm going to talk about some new work we're focusing on looking at biomarkers of phytoplankton in the Ross Sea region. Atmospheric processes and the composition over the Southern Ocean and those source regions influence ice core chemistry. So for those of us who work on impurities in ice cores, this is something we think about a lot of the time, is of course those processes and changing source regions drive the variability in the chemistry we see in ice cores. In ice cores, we can pretty much measure the full spectrum of aerosols over the Southern Ocean, from long-range transport from terrestrial regions such as mineral dust, volcanic ash, fire aerosol, and pollution aerosol, all the way through to marine aerosol, such as sea salts from the surface ocean, from sea ice environments, and also organic aerosol that's emitted by primary producers in the surface ocean. So the variability in ice core chemistry records are influenced by changing conditions at the source region, through to processes en route, the deposition regime at the ice core site, and in some cases even post-depositional processes whereby the original signal deposited to the snowpack is modified. And all of these, from the source region through to transport and deposition processes, are really important for us to understand in the modern day atmosphere in the development of new ice core proxies. So I'm going to talk about some new work we're focusing on phytoplankton proxies. And of course, why are we interested in developing ice core records of phytoplankton? Well, phytoplankton are really important because they form the base of the food chain. They're the main energy source for Antarctic marine life. They also play a really important role in the global carbon cycle through primary production, and that's the conversion of atmospheric carbon dioxide into organic matter via photosynthesis. And in this picture here, you can see of ocean color. This is of phytoplankton pigments seen from space. The Southern Ocean is very productive, and you can see around the coastal regions of Antarctica in red, very, very productive waters. So we have a few decades worth of satellite data about phytoplankton. Observations are pretty scarce. And so why do we need longer records of phytoplankton? So the first reason is to predict the response of phytoplankton to change. Half of the global marine primary production occurs in the Southern Ocean. We need to understand what drives that phytoplankton change in the past. The second reason is to help us constrain past sea ice conditions. If we're able to get a robust ice core proxy of diatoms or sea ice algae, then we can use that to reconstruct sea ice conditions in the past and to put in context the recent trends in sea ice in Antarctica. So this graph here you can see of mean annual Antarctic sea ice extent from satellites, and it's been slowly increasing in contrast to what's going on in the Arctic. In 2016, there was this dramatic drop in sea ice extent, as you can see on the picture of Antarctica here. And then since then, the sea ice extent has been increasing to previous levels. The ice core community has been working towards using biomarkers to constrain sea ice conditions, and there are a number of potential biomarkers that show really great promise. So of course, methyl sulfonic acid, MSA, this is very well established in the community. It's this biogenic emission from plankton growing in the ocean. And you can see here this correlation plot of winter sea ice conditions with MSA and an ice core from West Antarctica. A more recent biomarker that we've been working on is this fluorescent dissolved organic matter. The community's also been working on lipid biomarkers. And this figure here on the right is an example from Bouvet Island here in the sub-Antarctic. And we can see the correlation with MSA from the ice core and sea ice concentration downwind from the ice core site. And the second panel is a lipid biomarker. It's a fatty acid measured in the ice core, and we see this same nice correlation with sea ice. So we're particularly interested in looking at biomarkers and ice cores from the Ross Sea region. The Ross Sea region is one of the most productive areas in the Southern Ocean, and it's recently been designated the world's largest marine protected area. That area is shown here in white. And we're very fortunate that we have a whole suite of ice cores that surround the border of the Ross Sea Marine Protected Area. In the figure, just a couple shown that we have this great opportunity to use these ice cores to reconstruct past temporal and spatial variability using biomarkers of the two main phytoplankton blooms in the Ross Sea region. So there are two blooms in the Ross Sea. The first occurs in the Ross Sea Polinia, and the second occurs here in the southwestern Ross Sea, and both of these vary in terms of their composition in the oceanography. In the Ross Sea region, every summer as it gets warmer, the sea ice breaks up, it becomes warmer, and we get an influx of nutrients to the surface ocean. This triggers these vast phytoplankton blooms that can be seen from space. And these phytoplankton give off biogenic emissions. They give off organic nitrogen in the form of amines, biogenic sulfur, organic matter that we can measure as the fluorescent dissolved organic matter signal, and also some lipid biomarkers such as fatty acids. So we've been working on a few of these biomarkers from Roosevelt Island ice core from Rice in the eastern Ross Sea. And the first example is of biogenic sulfur. So this is very well established. Biogenic sulfur, this is one season from the ice core. And you can see this very strong signal in summer that we see in lots of Antarctica, which really parallels satellite pigments of chlorophyll. The second biomarker is based on fatty acids. And here's an example from summer snow at Rice. And we're able to detect a whole range of different fatty acids that are diagnostic of bacteria, diatoms, dinoflagellates, and algae, which is really exciting because we're able to start to look at the composition of the plankton in the ocean. Then the third example is this organic matter, and we're interested in looking at the fluorescence of this organic matter because that tells us about the microbial, gives us a microbial fingerprint based on different sources from terrestrial through to sea ice and open ocean phytoplankton. And what we see at Rice is a fluorescent dissolved organic matter signal consistent with phytoplankton growing in the open ocean. I'm going to talk a little bit more about this FDOM signal and how we can really use aerosol observations of biogenic emissions to help us understand these new proxies. So here's a nice example of aerosol observations made on a ship cruise from New Zealand. It went down into the Amundsen Sea. You can see on the here, it went from New Zealand down into the Amundsen Sea and then back further north to New Zealand. And this was a really great cruise because it went through both open ocean and sea ice environments. And when it made its way into the Amundsen Sea down here, we through the sea ice environment, we see this large increase in MSA in the atmosphere. At the same time, when the biology was dominated by diatoms. They also measured sodium and sulfate and other aerosols during the cruise. In addition to that, they measured this fluorescent dissolved organic matter signal. And that's really neat because this cruise passed through different conditions. We're able to see that signal in the FDOM. So the first signal on the left is able to characterize sea ice algae in the aerosol, and the second one is when the sea ice the cruise passed through open ocean, and we get this really nice signal here of phytoplankton growing in the open ocean. A third component was detected of a terrestrial-like signal. And these information is really fantastic because we're actually able to compare what we see in the aerosol to what we see in the ice core. And at Rice, we see this signature here of the open ocean phytoplankton. This signal is also seen in a fern core from Patriot Hills here in the Ellsworth Mountains. And this component here varies seasonally through time. Interestingly, different components were found in Waste Divide ice core. It did pick up the scumic-like terrestrial origin signal found in the modern day aerosol, but a couple of different components in the fluorescent dissolved organic matter were seen at Waste Divide. So we're starting to build up a bit of a picture of spatial variability around Antarctica, but also temporal variability. So to really understand what's going on in the modern day atmosphere and to help us interpret these ice core records, it's really crucial that we start to build up a bigger picture of aerosol observations in the Southern Ocean. And so that's what I'm going to focus the remaining few minutes of the talk on, is to talk about opportunities where we can collect modern day aerosol samples from both long-term monitoring stations and also future ship opportunities. So in terms of long-term monitoring sites, there are a couple in the Southern Ocean. The first is Cape Grim Baseline Air Pollution Station in northwest Tasmania. This is a very well-established site and it's sampling air from this sector of the Southern Ocean. It's sector controlled, and so it's looking at these really pristine air masses. The second one is Bering Head down the road. Again, this is sector controlled and we're looking at air masses from a different sector of the Southern Ocean. Both of these sites have an archive of filters, of historic filters, and so it's a really fantastic opportunity for us to go back and utilise these filters to start to measure some of these new biomarkers on them. In terms of ship-based sampling, there are a few opportunities coming up in the future. So the first is on the RV Tangaroa. It's heading down to the Ross Sea in January 23. At that same time, our Spanish collaborators are heading down to the Weddell Sea on a project called Polar Change. That's a large international program investigating emissions from the Weddell Sea, from sea ice and open water environments. In our Australian collaborators, there are a few opportunities over the next few years to go down to coastal East Antarctica and also into the marginal ice zone on the new Australian icebreaker. So how do we collect these aerosol samples? At a land-based station, we use a high-volume aerosol sampler. So this is an example with a PM10 size selective inlet. So it's collecting all aerosols less than 10 microns. We can deploy those on ships, as seen in the top photo, and even take them down to Antarctica. The second option on ships is if the ship has an aerosol, an air intake, like the investigator, that air can go down into the aerosol laboratory into a range of instruments and inline filters. So there are a few opportunities coming up over the next few years to collect aerosol samples, and anyone who's interested in sample sharing or working on new proxies, marine proxies, organic proxies, who would like to be involved in these projects, please do get in touch. So in conclusion, atmospheric processes in the source regions influence the aerosol composition above the Southern Ocean and hence the ice core chemistry. And we looked at the example of phytoplankton biomarkers and how getting an understanding of the sources and the transport processes to ice core sites are really important to interpret the longer term records and to validate these new proxies. And then lastly, there are a few future opportunities to collect aerosol samples at land-based sites and on ships in the next few years. Kia ora and thank you.
Thank you so much, Holly. Does anyone have any questions? In the meantime, I kind of have a bit of a general question, Holly. When it comes to sampling the aerosols at the site of the core or in Antarctica, as you were describing, how is it that works based on aerosols and the temperature? Are there some issues that might take place there?
So it's the aerosol sampler in Antarctica?
Yes.
Yeah. So yes, when we're looking at, we're interested in the air volume, and so we need to correct that for temperature and pressure, and that's something we do. Yeah, but we can run a pump and collect samples in those cold temperatures.
And in terms of maintenance, do you know of like how the maintenance of it would actually take place?
So quite often these are relatively short campaigns for a month or just the summer season. Yeah. Okay. Otherwise, stations like Neumeier and at Halley, they have dedicated aerosol or atmospheric stations, and so they have sort of an air inlet, and that air can go into a warm lab where they sample it.
Oh, that's great. So one last question on this. Oh, actually, Matthew, you have a question. I'll let you take over.
Hi, Holly. Great talk. The planned voyages, such as the Polar Project voyage into the Weddell Sea, do you know if they have any plans to collect fluorescent aerosol measurements like the Amundsen Sea voyage?
Hey, Matt. I think they're going to be doing fluorescence measurements in the ocean itself. We're hoping to collect filters to make those measurements. So yes, we can definitely talk about that. That would be great. And the other cool thing about that voyage is they're going to have a number of aerosol mass spectrometers on board. So they'll be measuring real-time atmospheric chemistry, which will be really valuable to help interpret those fluorescence measurements.
Yeah, brilliant. Exciting stuff.
Does anyone else have any questions? If not, thank you so much, Holly. That was very exciting. I'm looking forward to hearing what else comes out of that.